have revealed a basal dichotomy within vascular plant, separating eophyllophytes from lycophytes. Under eophyllophytes were spermatophytes and monilophytes. Monilophytes is a fern, and spermatophytes is a higher plant. Then I will focus on lycophytes, or we call it as Lycophyta. Okay. Lycophyta or lycophytes is a small and conspicuous group of plants today. They are oldest extant group of vascular plant. The most significant features of lycophytes are the leaf is very small and less than 1 cm. There are 3 families under Lycophyta, which are Lycopodiaceae, Selaginellaceae and Isoetaceae, and my research will focus on Selaginellaceae. Selaginellaceae have only one genus in this world, which is Selaginella. There are about 700 taxa from Selaginella worldwide, 29 taxa from Malaysia, and 9 of it were endemic species in Peninsular Malaysia only. Here are some characteristics of Selaginella. Selaginella is known as spikemose or fern-alike. The leaf is simple and scale-like, or known as microphyll. The leaf is less than 1 cm, consists only one vein, and arranged in alternate, opposite, and swirling. Here is the arrangement of the leaf. The small one at the middle of the branch is the median leaf, and the larger leaf here is the lateral leaf. Many species of Selaginellaceae have a very distinctive leaf arrangement with 2 rows of smaller leaf and 2 rows of larger leaf. Okay. The leaf of Selaginella, there are 6 kind of lateral leaf shape: ovate, ovate triangulate, ovate lanceolate, elliptic falcate, elliptic and oblong falcate. And here is the median leaf apices. Okay. The structure like a tone here is arista, and the median leaf with the arista we call it as apiculate, and the median leaf with arista we call it as aristate. The length of arista can be one of the characteristics to differentiate one species to another species. The spores in the form of strobilus, which located at the tips of the branch. Heterosporous is a Selaginella have 2 types of spora, which are megaspores and microspores, and on the other words we call it as heterosporas. Here is the structure of strobilus. We can see here the structure of ligule. The differences between Selaginellaceae and Lycopodiaceae is the presence of the ligule. In Lycopodiaceae, they have no ligule. Ligule. Here is the diagram of strobilus of Selaginella. There are 2 types of strobilus of Selaginella in Malaysia: tetragonous type and bilateral type. This characteristic is the most obvious character that can be used to differentiate one species to another species, even with the naked eyes, without using a microscope. Okay, Selaginella may be erect, sub-erect, prostrate, and scrambling. Branch system is a series of dichotomies and anisotomers, and may result to a sidopinnate array and a fan shape of flageolate array. Here is the diagram of branch system. There are 6 branch system in Selaginella. The first diagram is a cytopinate with a small axis simple. The example of the species is Selaginella wallichi. The second one is cytopinate with a smaller axis forking at tips, and the example of species is Selaginella stipulata. The third one is cytopinate with smaller axis dividing in cytopinate fashion. The example of the species are Selaginella wildenoi, Selaginella argentea, and Selaginella frondosa. The fourth one, cytopinate with smaller axis obviously dichotomous. The example of the species are Selaginella caulescens and Selaginella roxburghi. The fifth one, trabelate with few dichotomies. And the example of the species are Selaginella intermedia, Selaginella alutacea, and Selaginella mayeri. And the last one is trabelate with much dichotomies, and the example of the species are Selaginella purpurea and Selaginella plana. Okay, I go to anatomy part. This anatomical part have not done by me actually. It have been done by Hazwani 2011 if I'm not mistaken. Since they have a data from anatomical part, so I will just go through. The venation patterns in Selaginella is a single. Selaginella have only one vein. Only one vein. So no different for the venation pattern in Selaginella. The anatomy of stips. There are two group of stele in Selaginella, which are monostellic group and tristellic group. The three at the top here is the monostellic group, and 2 at the bottom is tristellic group. You can see the stele for the tristellic group. There are 3 stele here, and for the monostellic group only 1 stele at the middle. So the species for the A is Selaginella argentea, Selaginella intermedia variety delicocentrus, Selaginella intermedia variety intermedia, Selaginella morganis, Selaginella ornata, Selaginella polita, Selaginella roxburghi variety roxburghi. For the B, Selaginella alutacea, and C, Selaginella frondosa. For the tristellic group, for the diagram D, Selaginella mayeri, Selaginella plana, Selaginella stipulata, and Selaginella wallichi. And for E, Selaginella wildenoi. Okay, for the molecular part. For molecular part, the region that I've used is from chloroplast region and chloroplast genome and nuclear genome. The tree here is from neighbor joining analysis from the RBCL region. Okay, the number on the branch here known as bootstrap value. The higher bootstrap value means that the relationship between one species to another species was be very supported or very strong. Here is for neighbor joining analysis, the diagram. You can see the tree and the bootstrap value here was very higher, and most of them is 100. Okay, here is the diagram, is the tree topology for maximum parsimony analysis. I choose Huperzia lucidula and Lycopodiella senua as a outgroup for my family. And here is the maximum likelihood analysis. You can see the tree. Okay, here is the previous study in Selaginella. Wong have been done on morphological part. Chin 1998 preliminary study on anatomy of Selaginellaceae, and followed by Leisman 1961, Pariha 1965, Bierholtz 1971, and Ogura 1972. Buck and Lukansky 1976. Selaginellaceae has attracted botanical attention for many years because of its peculiar anatomy. Coral et al. 1999 and 2002 using RBCL region on 62 taxa of Selaginella. And Coral and Kendrick 2004 using 26 srDNA on 23 species of Selaginella from their previous study. Thank you.